All right. So, like Nirman said, my lab works on zombie ants, and we work on this because we're really interested in studying the mechanisms underlying behavioral manipulation by parasites of their animal hosts. Now, there are quite some parasites that are able to do this. However, because most of these parasites and both their hosts are not considered model systems, for a very long time, people have not really studied those mechanisms. So what I'm going to show you today is some of the work that we're doing in my lab. We're using Ophiocordyceps fungi that infect carpenter ants as a model system to study this phenomenon. And as you'll see, we're slowly moving this study trajectory from the descriptive to the more mechanistic. So I hope you can appreciate some of the things that we're trying to do. You'll also see that we use a combination of different biological techniques and biological fields. So our work is very integrative. So we work from a fungal molecular perspective, but we also use omics techniques, and we use animal behavior to answer some of the questions that we have. Now, to introduce you a little bit here. So carpenter ants of the foraging caste, they generally get infected by Ophiocordyceps as they pick up fungal spores during a foraging bout. Upon initial infection, the ant returns back to the ant nest where she first behaves normally, and then the infection initially remains unnoticed by nestmates. However, after a certain incubation period, the infected individual will leave the nest, not to precipitate in foraging activities. We found that they are actually not so good at this anymore, but what they do is they wander away from the nest to find a piece of vegetation to climb and latch onto with their mandibles. Once perched at this elevated position, the parasite quickly kills the ant and consumes her innards to generate enough energy to produce a fungal stalk that you see growing out of the ant on the right here, and a fruiting body from which the spores then will emerge that can infect new ants. Now, the biodiversity of Ophiocordyceps fungi infecting ants is very high. What we find is that we find this phenomenon in a lot of different places across the globe, and all the different ants that we see that get infected with Ophiocordyceps have their own specialized Ophiocordyceps species that has co-evolved with that host and is able to manipulate it. Now, depicted here are some of the places where I've done field work so far. Again, this is not the only area in the world. You can find this on the African continent as well. You can find this in Asia, not so much in Europe anymore because of anthropological effects, we think, but in the past it should have been there as well. And one thing that is striking, if you look at all these different ants that get manipulated in all these different places, is that it is highly stereotypical where they actually end up and what they bite, depending on the ecosystem you're in. So you'll find ants in the Atlantic rainforest biting the main veins of leaves, like is depicted here. Ants in the Amazon seem to be biting the edges near the tips of the leaves. And in contrast, you'll find ants in temperate forests, like the one up here from South Carolina here in the US, to actually bite twigs instead of leaves. Which kind of makes sense if you take into account that sometimes these cadavers, they need to overwinter until the next summer in order to transmit. So biting leaves would not be the most optimal there. So we see these very local adaptations in the climbing and biting behavior as well, which I think is incredibly interesting. Now, at the moment, most of our work is in Central Florida because we're lucky that we can find this system right on our doorstep. Actually, most of our field sites are 20 minutes from campus. And what we find is that the carpenter ant here, the Florida carpenter ant, which is incredibly abundant, is manipulated here. And we see that they mostly are biting Spanish moss, pine needles in this area. Now, this final climbing behavior of Ophiocordyceps-infected ants is also referred to as summit disease, which can be observed for quite a few insects beyond ants and that are affected by behavior manipulating parasites. So this can even go beyond fungi. In all cases, the summoning behavior is adaptive to the parasite, as it increases the chances of the parasite to successfully transmit to the next host. This transmission can, for instance, be the release of infective propagules like spores, or, for instance, trophic transmission. So what we see, for instance, beyond Ophiocordyceps is that Entomophora fungi, depicted here on the left, which are a completely different phylum from Ophiocordyceps, can infect a range of insect hosts and reside and basically introduce this similar summoning behavior. Additionally, increased propagule transmission, we also, for instance, find this for baculoviruses that infect moth caterpillars. However, in this case, the disease is generally referred to as treetop disease. Alternatively, as I said, you can also have trophic transmission. So we see this, for instance, for ants that get infected with dicocoelium. In this case, the ants are infected with a trematode that causes them to climb up a piece of grass and latch on to make it more likely that grazing cattle will consume the infected ants. This way, the trematode ends up in the gut of the grazing herbivore, so trophic transmission in this case, where it then can continue its life cycle. So these worms have a complex life cycle in which they need multiple different hosts to finish it. Now, there's also all sorts of, these are examples of some of the well-known and relatively well-studied parasite manipulation systems. But manipulative infections are discovered on a regular basis still, especially since people are now more aware of this phenomenon actually existing out there. So like this grasshopper, for instance, here, this was found here by undergraduate students from our entomology club here at UCF. However, in this case, we still have to verify what parasite actually caused this manipulation and what type of transmission might be taking place here. So lots of cool stuff still to discover beyond the model systems that we currently have. Now, my lab doesn't want to advance. 1 second, please. There we go. My lab basically tries to answer what are the mechanisms underlying behavioral manipulation. So we don't focus as much on the biodiversity. We leave that to some other experts in the field who are much better at this than we are. But we really ask questions about how does this work? So nowadays we use camponoti-floridani, so the Florida carpenter ant, and its specialized Ophiocordyceps camponoti-floridani fungus as a model system. So why do we think this is a really good model system? Because there's so many to choose from. Well, both organisms we can easily collect and keep in the lab. So over the years, we have fine-tuned the techniques to do this. So we're able to culture Ophiocordyceps now and have it in a form in which we can actually infect our ants. So as such, we also developed infection techniques and are step-by-step learning how to do this better and better. So our infection experiments are becoming more successful. What we've done over the years as well to really be able to kind of look underneath the hood and see what's going on is we sequenced the genomes of several Ophiocordyceps species and annotated them. And we're very lucky that camponoti-floridani is for the myrmecologists is considered a really interesting ant species. So that genome has been sequenced by our colleagues. So we're very lucky that we can take advantage of that in this case. And then what we're currently working on, and this is what we have this National Science Foundation money for, is to adopt functional genetics techniques for our fungus to really make it a model system in which we can knock out genes and do all sorts of functional studies, for instance, to really see which genes are playing a big role in these in the behavioral manipulation that we see in our so-called zombie ants. As such, our work is at the boundary of infectious disease and animal behavior. Like I said, I run a very integrative lab where we use a bunch of different techniques to answer our questions. And one of the things that we're also doing, and I've kind of skipped that bit, and I hope I don't talk too much and I get to that a little bit later in the talk, are to show you that our work is also at the boundary of infectious diseases and biological clocks, because we think biological clocks play a really important role here as well. So to answer the questions about the behavioral manipulation, we specifically ask which fungal effectors are secreted and involved in this process. So in that case, we really look at things from a fungal point of view, and from that we hope to also learn then which insect host pathways are affected by these fungal effectors. Now, at the boundaries of biological clocks and infectious diseases, we look at how the biological clocks of two organisms actually interact. So we ask questions about if you have a parasite and a host, how are they dealing with one another, and how are those clocks either clashing, or is one maybe hijacking the clock of the other host. And we specifically look at that in this system, as I'll show you that there are some really intricate timing aspects that make us think that this is an important thing to ask. And as I mentioned before, we use a bunch of different techniques all together to kind of try and answer these questions. Now, when we're talking about the first question, then what are the mechanisms underlying behavioral manipulation, and why are we thinking about effectors? One of the things that we think is happening here is that the ant's brain tissue or nervous tissue doesn't get degraded necessarily by the fungus, as we know from some histology work done by others. So David Hughes' lab has focused on this quite a bit, both with more classic histology techniques and newer, fancier techniques. But the answers that they're finding are still the same, is that as we look at an ant, so this picture here is to kind of orient you. This is an ant head, its brain is right here, its mandibles are right here, and this is what most of its head normally would look like if the ant was healthy. So what you see here is a densely packed muscles that help this ant with its by using its mandibles, and this is really like these are really strong muscles. These ants can really latch on very well with those mandibles. But what we see in an infection is that while the brain stays relatively intact, we see that there's heavily degraded muscle tissue, and that the openings that are then there are all invaded by fungal blastospores. So these are the yeast-like cells that Ophiocordyceps produces when it's inside a healthy ant. So this makes us think that since the fungus is not invading the brain in such a way that it's degrading that or really mechanically affecting that brain tissue, we work from the hypothesis that, I'm sorry, my computer is again not letting me advance. We work from this hypothesis that very likely the fungal parasite is secreting some sort of effectors or bioactive compounds, if you will, that affect the ant behavior. Now we look at this question from multiple angles. So as you can see here, we use multiple omics techniques to look at this. And this is giving us some new hypotheses about some of the genes and compounds that might be involved in this process. And then what we're doing right now is we're moving on to functional genetics assays to test those new hypotheses. Now, I don't have time to talk about all these things, so I'm going to show you a little bit about the transcriptomics work that we have done that have led to those hypotheses. So all of our studies basically start with this, with us doing micro injections in our ants, either injecting those ants with fungal tissue or injecting them with a media in which we normally would keep and grow that fungal tissue as a control. So these are our sham infections. And what we then do is we put them in a behavioral setup, score these ants for their behavior, and sample them based on their behavioral phenotypes. So in the case of manipulation, the ultimate biting behavior that we see in the climbing behavior, again summarized as summiting behavior. If we see that happening, we can sample the ants that show this behavior, look at their gene expression, for instance. So that's an experiment that we have done across two different ant and fungal species, the carpenter ant from South Carolina, camponoti-floridani, and its specific Ophiocordyceps, which is called Ophiocordyceps-infected, and as well as the Florida species. Now, what we're doing in these essays is that we sample the ants based on their behavior. So we sample live manipulated ants to know what the gene expression is at that specific time point. Then we also sample ants that were manipulated but had died right after. So we get them right at that point. Then we have live ants that are healthy, that are sampled at exactly the same time of day as a control for what's happening to the ant genes. And we have fungal cultures that we use as a fungal control to contrast the gene expression in the ant against. Then we map those reads to our respective genomes. So in the case of the infected ants, we have mixed transcriptomes. So having both the Ophiocordyceps and the camponotus genomes is helping us to separate those reads and look at gene expression in both organisms. And then we do a whole bunch of analyses after normalizing the data to get the gene expression. So we use, for instance, principal component analyses, or PCAs. We use weighted gene co-expression network analyses, or WGCNAs, and the classic differential gene expression analyses, or GDGs. And in this case, what we did is we only looked at genes, or we only picked genes that were found across those various independent analyses and across the two independent fungus and species interactions that we did this experiment for. So only those genes we consider now to be potential candidate genes and pathways involved in the manipulated summiting phenotype. So to kind of give you an idea of what some of that data might look like, so for instance, the WGCNA that we did was really helpful because what we did is we made modules of fungal genes, and those modules are based on the expression values of those genes and kind of groups them together. So modules exist that kind of show the network of genes that kind of have a similar type of expression. We found a bunch more modules, but this is kind of a cropped version of that figure here to not overload you with a lot of unnecessary information. And we did that we did that for the ant as well. So since we have the gene expression for both. And then we tried to see if there was a correlation between some of the fungal modules that we found and some of the ant modules that we found. And we did see that there were two ant modules that were connected to two of the fungal gene expression modules. And those are depicted here. What we did then is we went into those modules and looked at which genes were there for both the ants and the fungi, and did an enrichment analysis to see which type of gene functions would show up as being enriched. Now, what we found is that neuronal maintenance and development, olfaction, memory, and circadian rhythms and light sensing were overrepresented on the ant side of things, and that they were connected in the network to fungal genes that are actually secreted. So we found an overrepresentation for genes that all have a secretion signal, so are predicted to be secreted. And that kind of makes sense if you are looking for interactions between the host and this fungal parasites that lives inside this host. Some of the genes that we found in there, I will discuss with you in a minute. So, but beyond this, what we found is that things are a whole lot more complicated, of course. So one of the reasons why we don't really understand the genetic basis of behavior in a lot of detail is because often lots of different genes and pathways and compounds are involved. So this is also basically the message that this exercise gave us, that a lot of different pathways in the ants, which are indicated in orange here, are affected during manipulation, and that quite some different genes in the fungi that are indicated in blue here might be related to those. So obviously, I don't have the time to go over all of them with you all. If you are interested, I encourage you to have a look at our paper. But to kind of give you just a general overview and some examples. So what we found for the fungal side of things here is that we found that both novel and unique fungal compounds with unknown function, so your small secreted proteins, if you will, but also some larger enzymes that haven't really been studied for fungi so much, we found in popping up in this analysis. We found compounds with known function in other manipulating parasites, so we could compare that. And we found compounds with known function in other fungi. So to give you an example of all three of those categories. So one of the things that we found was that these Ophiocordyceps fungi harbor a lot of different genes that encode potentially for secreted enterotoxins. So this is based on functional domains in those gene sequences. And what we know from enterotoxins is actually all known from bacteria that produce them. So these haven't been heavily studied or really been reported on a lot in fungi. And as we look at, for instance, the genomes of plant-infecting fungi, we don't really find any enterotoxins there. So while animal-infecting fungi seem to have these, plant-infecting fungi generally do not. So this might have been one of the reasons why these have not been studied for fungi yet. But what we know for bacteria is that these enterotoxins, they interfere with G protein-coupled receptors, which could lead to an accumulation of cyclic AMP and then dysregulated cell signaling and other processes. What we know, for instance, for bacterial enterotoxins in insects is that they are known there to be able to cross the blood-brain barrier. So potentially they could have an influence on brain tissue here. They dysregulate pheromone production, and they could eventually kill insects. Now, in our study, what we found is that there are 7 of these upregulated, and Ophiocordyceps has a lot of different genes that have enterotoxin functional domains, more than any of the other entomopathogens that we were able to look at in terms of their searching their genomes. So 7 of these are upregulated at manipulation, and there's specifically one that was upregulated really highly in both the species pairs that we looked at, so both Ophiocordyceps from South Carolina and from Florida. And we found them, they keep on popping up across all the different independent analyses that we do. So this is one of the candidates that we're really, really interested in. To give you an example of a compound that has a known function in other manipulating parasites, PTP, for instance. So this actually has been studied for that baculovirus that I mentioned before that infects caterpillars and causes summit disease there. What has been studied there is that actually this gene is important for enhanced locomotion activity in those caterpillars, which we normally only see during metamorphosis. And what we see is for the Ophiocordyceps transcriptome is that there is a 100-fold upregulation during manipulation. There's 2 more PTP genes that are significantly overexpressed, and we find that actually camponotus, in camponotus, the PTP genes that it has itself are upregulated as well. Now, additionally, when we look at the behavior, we see that enhanced locomotion activity is also seen in our infected camponotus. And so this might be an indication that maybe some of those summit-inducing parasites have some similar mechanisms going on to manipulate their host. So this is one of the other candidates that we're incredibly interested in. And then to give you an example of one of the compounds with a known function in other fungi, we found, so when we look at secondary metabolite clusters in the genome of our Ophiocordyceps species, for both the Ophiocordyceps that we looked at, we saw that there's especially, sorry, for some reason this went wrong. There we go. So for one of them, we see that this one is extensively upregulated during manipulation, and then it starts to be downregulated afterwards again. And we see this both for the Florida species and the South Carolina species. And when we look at that secondary metabolite cluster, we see that they they share this secondary metabolite cluster in quite some detail. So there's they both have a pretty similar organization. And when we further looked into it to try and functionally annotate it, we noticed that quite some genes are in there that we actually know from Aspergillus flavus. So what we think is happening here is that this secondary metabolite cluster is producing something that is very similar or might even be exactly aflatrim. We're trying to figure this out with metabolomics at the moment, what exact compound is being produced. But what we know from aflatrim is that it is known to cause Stargardt disease in mammals, and that this is due to the dysregulation of GABA processes and potassium channels, which then leads to muscle tremors and activity levels that change and confusion in those animals. Now, not to say, of course, that our insects are mammals, but maybe you can appreciate from these videos here that these healthy ants on the left move quite differently from the infected individual on the right. That definitely shows a lot of stumbling going on. Now, of course, these are all things that are hypothetical at the moment, right? So we only have our transcriptomics data that gives us these patterns, and from that we selected our candidate genes that we think are important. We also did this for the ant side of things, but I'm not going to talk about that today. I kind of focused on the fungal side here. But what we're doing now next is that we're trying to see which of these compounds are really essential to this manipulation process, to learn more about their exact function that is now inferred from homology through our annotations. So what we're developing at the moment is an Ophiocordyceps knockout system. So we're tackling this through two different approaches, both homologous recombination and using CRISPR. We're doing heterologous expression as well in a fungus that is fairly related, so Bavaria bassiana, which is in a different family, but we know that this is a generalist fungus that we can actually use to infect our ants, and doesn't have it doesn't really induce a change behavior so much that we would call it manipulated. So we are trying to heterologously express some of the genes to see if that has any phenotypical effect. And then we're doing yeast to hybrid to see if we can find any binding properties that might be interesting to look at, to learn a little bit more about how the fungal proteins might be interacting with receptors or other aspects in the ant host. And we're using protein production systems to kind of be able to apply proteins to our ants or to other animals to kind of see if there is just providing them with this protein might induce a phenotypic change. And to give you an idea of some of the stuff that we're doing. So William Beckerson is a postdoc in my lab who is playing with this right now. So since we can buy aflatrim itself, so the actual compound, of course, again, we don't know if our fungus exactly produces the same, but some of the pilot tests that we're doing, so this is very, very preliminary, we do see that stumbling effect in our ants that get treated with aflatrim, which resembles somewhat the behavior that we see in the infected individuals. So this is just to see how we could do those type of behavioral experiments. Now let me see how I'm doing on time. All right, I have a little bit of time left, so that's great. So what I want to do with the rest of that time is to step away from the neuromodulator hypothesis a little bit and talk about the clock hypothesis that we have, because the neuromodulator hypothesis does not cover the entire story. So one of the things that I left out of the life cycle here is that I explained you briefly in the beginning, is that biting behavior and this climbing doesn't just happen at any time of day. It seems to be synchronized at solar noon. So this is work done by David Hughes in the past in ants in that get infected in Thailand. So here, these are arboreal species, but the infected ants, what they seem to be doing is coming down to the forest floor. So what they did there is they followed those ants that would come down to the forest floor and see where they end up, and kind of record the time of day when they would actually show this biting behavior. And when they plotted it against solar elevation, they found that this happened around solar noon. Now, when we look at other systems, so the one in South Carolina that I worked on, and the one here in Florida, we see that in nature, at least for Florida, we kind of opportunistically observed this. So we didn't do such a detailed work because it's a little bit more difficult here to do that. But we did find any ants that we found in the process of this biting and still alive would always be somewhere around solar noon in nature. And what we see in the lab is, when we move things in the lab for infection studies, is that we also see the synchronization. However, in that case, it happens at a different time of day. We think that is because the environmental factors that would set the cues for what time of day it is, we can't exactly replicate it in the lab. So we think there's some sort of phase shift going on here. Interestingly, some field work that my collaborators did in Brazil showed that maybe this biting around solar noon has a really important function, because what they did in their studies is they looked at dense areas with a dense amount of these dead ants, and they would shade half of those areas and keep the other half as a control, so unshaded. And what they found was that most infected ants in those areas would show up in the control area, so where they didn't manipulate the light. So making us think that light is actually important in that final phase of manipulation, that is, ants might actually be attracted to it or seeking certain light levels. So being able to do this, during the daytime, while lots of these ant species that get infected are actually normally nocturnal, so their activity for foraging is mostly during the nighttime. So now this happens probably during the daytime because they need to seek out certain light levels. And it seems that this has a fitness effect on the fungus, because the ants that end up in the control area were much more likely to actually produce a fruiting body and therefore to transmit to other ants, while the ones that ended up in the shaded areas were less likely to produce one. So we think that this timing is actually functional for the fungus. Additionally, what we're finding in some of the lab work that we've been doing that was purely behavioral, so looking at how do infected ants actually behave at the beginning of the infection, and this was done by a master's student in my lab, Tenten Trin. So what we found, indicated in red here, is that those infected individuals kind of lose their daily rhythm, while the healthy controls have this very nice nocturnal activity that we expect for this species. So in this case, what we recorded was foraging behavior, so leaving the nest to go do all sorts of things, including foraging activity, because these infected individuals, what we also found in that study is that they're not that great at foraging anymore. They actually pretty aimlessly run around the setup. So we think there's a loss of rhythmicity there, and that might be related to some of the things that we found in our transcriptomics study, where circadian clock related genes actually stood out as being differentially expressed during manipulations versus control. So that's on the ant side of things. So what do we think is going on here? Well, organisms, as far as we know, all organisms have evolved biological clocks to anticipate predictable 24-hour changes in their environment. And they keep time through some sort of core transcription translation feedback loop. What is indicated here is the feedback loop for generally known for insects. Now, this feedback loop is entrained by cues from the environment, which are called zeitgebers. So this can be light, temperature, food intake, availability of food for predators, for instance, or so biotic factors can play a role here as well. Generally, light is one of the biggest zeitgebers. And this then, this feedback loop then gives rise to clock-controlled rhythms that can give rise to chronotypes. So individuals of a certain species don't all have exactly the same rhythm. It can be slightly different, and also throughout an animal's life cycle. Or different life stage. When you compare different life stages, they can also have different chronotypes. So, for instance, to take us as humans as an example, we are generally wanting to stay up later at night as teenagers and young adults. However, when we get older, we become earlier. So we get these earlier chronotypes, or we would like to go to bed a little bit earlier than we did as when we were younger and still going out to party, maybe. So all of these rhythms, they're slightly plastic, and as such, these rhythms aren't just there. They give their output can be behavior, like I told in my example, but also metabolism is controlled by this, immunity, physiology, and so on. So this leads to these daily rhythms in these different types of phenotypes. Now, what we're thinking can be going on here is that when you have a parasite with its own clock, it might be able to actually break into this process at any point. So we don't really know where this is, if this is at the core clock, or is it that this is at the output of the core clock, or the output of the clock, some of the clock controlled aspects. So we don't know that, but what we think is going on, at least for Ophiocordyceps, is that it is making use of plasticity in animal behavior that is given rise to by the circadian clock, and that it can use this as a prerequisite for manipulation. So this is a hypothesis that we're working from in my lab at the moment. I showed you some of the field studies and some behavioral studies that we're doing, but what we really need to do, because this is all circumstantial evidence, of course, is do the actual chronobiology experiments. Now, since we're not working with model systems for clocks at all, so for fungi, for instance, Neurospora is a model system for studying biological clocks, and for insects, Drosophila is often used. So what we first had to do is establish some clock baselines and answer some preliminary questions first before we could go and answer this question: what happens to the host clock upon infection? So the first question that we asked is basically, does Ophiocordyceps have a light entrainable clock? Because that would be important for it to be able to keep time that way. And that drives daily rhythms in gene expression and effector secretion. And from the transcriptomics work that we did, so basically what we did is we sampled Ophiocordyceps cultures every 4 hours and then looked throughout a 24-hour day. We did this for 2 consecutive days and then looked at the gene expression and looked at which genes would be rhythmically expressed. And we found that indeed it has rhythmically expressed genes. We did indeed find some core clock genes that we would expect to be there. And interestingly, we found that there's a group of day active genes and night active genes, and that, for instance, those are enriched for different types of gene functions. So the ones that have to do with pathogenicity and parasite-host interactions were actually showing up as enriched during the night peaking ones. So it does seem that Ophiocordyceps is able to keep time. So that's great. But then what happens on the host side of things? So does camponoti have a light entrainable clock that is plastic enough to give rise to different behavioral phenotypes? Because if something is very rigid, then it's maybe unlikely for this parasite to be able to break into it. But if it's somewhat plastic, that's important for survival for that organism, of course, but it also might function as an Achilles heel for parasites that come in and try to take advantage of that host. So we see from older behavioral work in ants that they do have these different chronotypes. So when we look at younger ants, they are generally in what we call the nurse caste. So these are ants that all kind of work around the clock within the nest. They never leave the nest to take care of the queen and the brood within the colony, so within their society. But as they get older, those ants are moving into what we call the forager caste. So these are the older individuals that generally go and forage for food. So they are going to go do these really dangerous tasks of leaving the nest and trying to find food sources. And they do this with a daily rhythm, as I showed before. So for nocturnal species, they do this mostly, for instance, during the nighttime. So that would make us think that they are indeed pretty plastic in their clock, but what we had to do is kind of show that there are some clock aspects that underlie this division. And this is work that Biplabendhu, who is a PhD student in my lab, did. So we did a similar experiment for the ants, like I just explained for the fungus, where we sampled ants, in this case, every 2 hours over a 24-hour day. And we did this both for nurses and for foragers. And we found that foragers have a lot more genes that are rhythmically expressed across a 24-hour scale. So they are show diurnal rhythms, where some of them are a day expression and some of them have night expression. And for nurses, we found that they have a whole lot less. So there's definitely something going on there that is different between those castes. Additionally, we found that nurses aren't therefore more arrhythmic. They actually had quite a few genes that had 24-hour rhythms in foragers that showed 8-hour rhythms in nurses. So what we think is that they keep time just slightly differently. So they have these 8-hour rhythms that kind of keep them in tune with a 24-hour day, but in a different way, obviously. And this might allow nurses to quickly change to become part of the forager caste when they need to, or foragers to switch back to the nurse caste if they need to, because this is something that can happen as well. So even though there is this division, when the colony really needs more nurses or needs more foragers, there can be this quick switch, which is generally indicated through social interactions and therefore the social environment. So it looks like that's, that second prerequisite that we had, that, the clock of the host needs to be fairly plastic, that is probably true as well. So now we can finally look at what happens to the host clock upon infection. So that's something that Roos Browns, who is a visiting master student from Utrecht University in my lab, is working on together with Biplabendhu at the moment. All right, so I ran out of time. So to kind of wrap this up, what we are trying to do mainly in my lab is to understand how fungi can manipulate ant behavior. And I showed you today two of the main hypotheses that we work from and some of the integrative work that we're doing to discover the biologically active compounds, which genes are affected in the insect. So that gives us also a genetic basis for insect behavior. Didn't talk too much about that today. I kind of focused on the fungal side of things. And we're trying to discover or trying to figure out how biological clocks might intersect here with this infectious disease as well. Now, for future applications, we hope that, if we learn how to break into ant behavior, we can use that as part of pest control or conservation efforts. Since we're looking into compound production in these fungi that have been hardly studied to this level, we find all sorts of new new compounds or novel compounds that might lead to novel drug discovery, or the last one I didn't discuss because I had to skip that part in the interest of time. All right, so these are obviously all the work that I discussed today is not just done by me. So the work I mainly showed was from William, Biplabendhu, Ian, and Tentane in my lab, who are all grad students in my lab or recently graduated, which Tintin did, but there's lots of undergrads that help us with this work as well. And we have lots of collaborators that help us with the things that, we're not necessarily experts on ourselves. So this is the fun thing about working across boundaries of different fields, that you get to interact with lots of people with lots of really interesting expertises. All right, I want to thank you for your attention. I'm sorry that it took me a little bit longer, but I hope you found it interesting. And if you have any questions, then I'm happy to take them.
I hope you can hear the applauses from this side, Charissa. Thank you so much for your exciting and very interesting presentation. And you can already see that we have some questions on the chat box, and I think Nikki's hand I see. So I will actually first give it to Nikki to ask her question, and then I will read the question to you from the chat box. Nikki, can you please?
Hi, thank you so much for your talk. It was super great. So I'm a plant biologist, so I might be a little bit off on some of this, but I did spend some time at Stacey Harmer's lab in UC Davis working on plant clocks. And I seem to remember that insect and animal clocks, they have a master clock that regulates the other clocks in the other tissues. And that's situated in a few cells in the brain, as far as I remember. And so it's quite interesting that some of the genes you found are genes that can cross the blood-brain system. Do you think that those are the genes manipulating the master clock, or how do we see that fitting together?
Yeah. So as I indicated in that schematic, as I was kind of explaining, in one slide, trying to explain the whole chronobiology field, which is hard. But I actually, if I would have to guess right now, and because we just got some of that data where we're looking at what happens in an infected individual, I would guess that the core clock is too robust for this parasite to break into. So what I'm thinking is that some of the clock control genes and those pathways are actually the ones that are being corrupted. So I would think that not necessarily the central nervous system related cells are the ones that are really altered here. I would be surprised if that would be the case, because that needs to be something that is pretty robust, like can have some flexibility, and it should have some flexibility, because otherwise, we couldn't travel to the other end of the world and get over our jet lags. But I think that's something that might be a bridge too far for a parasite to corrupt. So I think it's somewhere further down the line, and maybe specific pathways that are more behavioral, that are driven by the clock eventually.
Okay, thank you.
Sure.
And the next question is from Jeremy Ellison. And Jeremy is asking, do ants ever recover from exposure infection? And if so, what factors influence rate of recovery?
Yeah, that's a really good question. We actually think that they don't. So what we see in our lab experiments is that only a very small percentage doesn't, succumb to the disease in the end. And we think that has more to do with us maybe not having done the injections perfectly in those individuals than them actually kicking the disease. So especially for the worker ants, we think that once you get infected by Ophiocordyceps and it's really able to invade you, then that's about it for those individuals. In the past, I did experiment with the different castes, because of course you also have queens, you have the giants, so the new queens, if you will, that leave the nest to mate, and the males. And what we found is that those new queens, they actually are not too phased by getting injected. Now, I must say that back then I didn't really keep dosage in mind, so they're much bigger. So maybe I gave them too little fungus for them to actually be affected by it. But that's the only group that I've seen just not being faced by this infection at all, while all worker ants generally don't make it after infection.
Very, very interesting, like Jeremy said, but thanks, Charissa. And the other question is from Carlos Rodas. He's from Colombia, and he's also saying that congratulations for great presentation. Do you think we can implement something similar to your research with other genus of ants such as Atta and Agromymetes that are important forest pests in Central and South America?
Yeah, that's a really good question, and it's something that I think about a lot. So to go and directly implement Ophiocordyceps for this type of pest control, or and infections in other ant species, I think is not very feasible because the fungus is so specific. So one of the things that I didn't show today was me trying to do common garden experiments where I took the fungus and I infected other species as well that are sympatric species from the same region, even from the same genus. And if they don't normally get infected with Ophiocordyceps, they don't show this manipulation. They did die because I kind of forced the infection there, but in nature we also don't ever find them infected. So this fungus being so super species specific, I don't think you can just directly take Ophiocordyceps and put it on Acromyrmex, for instance. However, if we happen to find that it has its own fungi that affect it in some way, then of course we could do some interesting stuff with that. The other thing that we're thinking about is, and this is part of the heterologous work that we're doing, is maybe some of the compounds that we find produced by Ophiocordyceps, we could maybe implement in some ways, either by having other fungi produce them or by using those compounds in pest control or in experiments with other ants.
Thank you, Charissa. And actually, I will not ask a question directly, but I want to take a few minutes because we still have some time to explain you what you explained to me earlier about your science outreach. So if you can just give us what is the next step for you for the science communication and outreach you are doing. I think there are many mycologists and entomologists that will be maybe interested to do something similar that what you are doing.
Yeah, so one of the cool things about the funding that we currently have is that it also gives us opportunities to do really impactful outreach. So one of the things that we're developing at the moment is a virtual reality game in which we kind of introduce this infectious disease system that we work on and kind of make it available to the public in a fun, gamified way. So what we're working on at the moment is basically a zombie ant game where you are in the game, you are the fungus, and you have to make different decisions about, how to use resources from your host and when to manipulate it and how to interact with other individuals and so on. So we take aspects from our research and we put that in the game. And this is something that once it's done, so once we have developed it, and I'm developing it together with students from UCF. So some of the students that are in our game design program, they're actually the ones making it. We're going to make this freely available online to everyone. So so schools and museums and so on can use this. So we hope to kind of get people excited about fungi and insects and research in general in that way.
And definitely you will make me very excited. I'm looking forward to the game. So I think Tuan has a question.
Charissa, Tuan here at FADI, so thank you for a fascinating talk. So since the relationship between the fungi and the arm are so specific, and also like you mentioned, almost all of them die in infection, I'm wondering regarding the evolution process, is it a one way? Does the ant get anything out of this? If you look about it, does the ant got any benefit from being infected in the long term? In terms of evolutionary, because why is it not being wiped out or going extinct? Because there is no the specificity on one hand, and the other one is almost like you mentioned, there's no resistance, so they then die at the end. So
yeah. So this is a really great question. So we don't think the ants get anything out of it, really. So it's really, I would say it's really purely parasitic. However, this fungus plays a pretty clever game. So it doesn't wipe out entire colonies. So that's one thing. So there's not a large, a really big selection pressure here to get rid of that fungus. Additionally, this fungus is only affecting the forager caste, so the ants that go out to get the food. They're the older individuals that are more likely to succumb because of old age anyways, or because of their foraging bouts. So foraging is a very dangerous task. You might not make it back home because, you get eaten, you don't find the way back, you get desiccated, things like this. So there's all sorts of other issues by which that forager caste dies. So it's not just the fungus. So we think that this is kind of what keeps this fungus going and keeps the ants from really recognizing it in such a way, and there being a really strong selection pressure against this disease. So the way I try to describe it to people always is it's more like a chronic cold. It's there, it picks up some ants from the colony, but it's not really putting a lot of pressure on that colony. So even though individuals die in the end, there's still plenty of individuals left for this fungus to keep on infecting.
All right. Thank you.
Thank you, Charissa. I think if there is no other question, we can maybe close the session, but I can really say that Charissa is quite active on Twitter as well, and we will also share her email address that you can find, so you can ask the questions anytime to her. But Charissa, thank you so much again for joining. It was great for us, and I will just hand it over to our director, Bernard Silliper, to close the session.
So, yeah, thank you from my side as well. I think, can everybody hear me? I'm sitting here in the same room as Erin, I'm just a little bit behind the house. Sort of put my camera on here. Yeah, it's been absolutely fascinating. I knew it would be fascinating just from the talk itself, but the depth that you've given to it really is interesting work that I'm looking forward to learn more about and to see developing over the coming years. So all the very best for your work. Thanks for spending this time with us. We look forward to learning from you when you visit us at some point. We hope that is soon. So thank you very much for the time and a great talk. All the best.